Hello everyone and welcome to our session dedicated to the basics of cell signaling. As usual, I'm gonna recap briefly what we have learned in the last session dealing with cell biology topics. First of all, I reminded you that units and unit conversions are going to be very important for the mPharm program. So if you haven't done so, please do have a look and make sure that you understand what certain units actually mean and how to convert them. We have also learned that cells are quite small. A usual cell is around 10 micrometers in humans, and bacteria are 10 times smaller, with most bacteria being around 1 micrometer small. Viruses are even smaller. They are below the diffraction barrier of the light, and you will need very specific types of microscopes in order to resolve them. For example, electron microscope, because the size is merely 100 nanometers in average. We have also learned how most cells actually look like. So the first important element of the cell we had looked at were plasma membranes. And plasma membranes are composed of phospholipids. We have some cholesterols, protein, and also other components. Next important component of the cell is the cytoplasm. And the cytoplasm is underneath the cell membrane. And this is basically a schematic view of a cell. We have the plasma membrane, and all of this stuff in here is surrounded by the cytoplasm. The components of the cytoplasm are obviously the cytoskeleton. Those are interconnected filaments and fibers. We have the liquid compounds of the cytoplasm, the cytosol, and we have the organelles. This does not include the nucleus. The nucleus itself is an organelle that contains the cell's DNA, and it is more or less the control center. The ribosomes are organelles in which amino acids are hooked together to make proteins. The Golgi complex is in charge of processing and transporting protein and other materials out of the cell. And we have the endoplasmic reticulum. This is the organelle that makes lipids, breaks down drugs and other substances, and packages proteins for the Golgi complex. We have also learned that there are at least two types of endoplasmic reticulum within the cell. We have the smooth endoplasmic reticulum, and we have the rough endoplasmic reticulum. The difference, the most obvious difference between those two types of reticulum is that the rough endoplasmic reticulum does contain ribosomes. In contrast, there are no ribosomes on the smooth reticulum. Another important organelle within our cells are mitochondria. And mitochondria are the organelles that break down food molecules to produce energy in form of ATP. In addition to this, mitochondria also do produce heat, but this is something Dr. Cottrell will talk about in one of his biochemistry lectures. Last but not least, there are also the lysosomes, and those are the organelles that digest food particles, waste products, and cell parts. So what are the learning outcomes for today? First of all, you should appreciate the necessity of cellular signaling. Moreover, you should be able to differentiate between paracrine, endocrine, and autocrine signaling. You should be also able to explain the terms receptor, ligand, and signal transduction. You should know the three phases of signal transduction and explain the processes occurring during each of those. Moreover, you should be able to describe how signals are transduced from the cell surface receptors to regulate the transcription of genes. So, why do cells need to communicate and respond to the environment? Simple answer is they need to do it in order to survive, and when appropriate to replicate. And this is critical that the cell is actually able to recognize what's going on outside of the cells. Cell signaling evolved actually quite early in the history of life. So all of the cells you can imagine are actually able to react to certain signals. And the ability to signal has become more complex as life increased in complexity. So signals received may originate from other cells in the surroundings of the cell, or actually even far away. It can actually be a result of changes in the physical environment and can take various forms. So we can have signals produced by other cells, and we have also physical and chemical signals. For example, increase of temperature will activate certain biological response in the cell. However, the most common type of cellular communication involves exchange of chemical signals. So how do prokaryotes like bacteria respond to signals? Importantly, bacteria respond primarily to environmental changes. They do have certain quite primitive ways to react to presence or signals released from other bacterial cells, but they react mainly to changes in the environment. They respond to changing temperature, they can respond to osmotic stress, the presence or absence of nutrients, and so on. And they do so by moving towards or away from the stimulus. And this is usually controlled by the motion of flagella. Also, the synthesis of new proteins to optimize survival under prevailing conditions may be the answer of a bacterium to change in environmental cues. For example, if there is glucose as food source in the medium, obviously bacteria need to have the enzymes that are responsible for breaking it down active. So whenever bacteria sense signals, they activate gene expression, resulting in synthesis of enzymes to use a new sugar source. So let us have a look at slightly more complex cells. Here I would like to talk very briefly about another way to talk to another signals that are exchanged in very primitive unicellular organisms. In this case, I'm talking about yeast. Those cells use exchange of chemical signals to find and attract suitable mating partner. They are able to sense what the phenotype of the other yeast cell in the surrounding actually is, and if this cell is actually suitable for mating. And to do so, they are using short peptide called mating factors. But this is only additional information, because as usual, our main focus are multicellular organisms, especially the human body. So multicellular organisms respond to both physical environment and also to the presence of other cells. The cells within such multicellular organisms need to know that they are in the correct place, and this is the first piece of information. And the types of stimuli our cells are able to respond to are, for example, growth factors. Those are cell matrix contact, hormones. We can have light. So, for example, certain cells in our eye, in our eye, are able to react to presence of photons with certain biochemical reactions. Our cells can also react to cell-to-cell contacts, odorants, and this is quite important for our sense of smell, and also to touch. Our cells are able to react to the pressure and also surface topology. Lastly, some of our cells are able to react to temperature. They can adapt if the temperature drops or increases. The responses occurring after sensing a stimulus are usually more complex and diverse and control both behavior of individual cells and also coordination of cellular responses in a tissue or even in the whole body. So, what is signal transduction in this context? Signal transduction is the mechanism by which a cell is able to respond or adapt to changes in its environment. So why do we need to know about signal transduction, or why do you as future pharmacists need to know it? You need to understand signal transduction to be able to understand how cells work. And also, you need to know how signal transduction works to understand and treat diseases and disorders, because many diseases are a result of failure of signaling pathways. And one very good example is cancer. In cancer, cells are not longer controlled and proliferate without the presence of certain factors that usually induce division in our healthy cells. And indeed, this is well recognized and widely accepted in the literature that signal transduction is one of very, very important contributing factors to in development and progression of cancer. With this brief overview, I would like to end session number 1, and I will see you in session number 2, where we are going to be talking about different type of cellular communication in the human body. Thank you very much for your attention.